For any medical student, antibiotics can feel like trying to climb a mountain in the dark. But what if you had a map, a system, a logical framework to make sense of all of it? Well, that's exactly what we're going to build today. Let's dive in. Okay, let's be honest. Have you ever looked at a list of drug names, penicillins, cephalosporins, tetracyclines, and just felt your head spin? If you have, you are definitely not alone. The sheer number of these drugs is staggering, and just trying to memorize them all is a recipe for burnout.
So, we're throwing out rote memorization. Instead, we're going to build a mental framework. Think of it like a filing system for your brain that will let you take any antibiotic, whether it's brand new or decades old, and immediately figure out its place, its purpose, and its properties.
And the foundation of our entire framework, the very first question we always ask, is the most fundamental one. How does this thing actually work? Everything else we learn about an antibiotic is built right on top of this.
I want you to think about it like you're attacking a medieval fortress. Are you going to smash down the walls? Are you going to cut off the supply lines? Or are you going to sneak inside and shut down the weapons factories? This mode of attack is what we call the mechanism of action, and it's the absolute most crucial way to classify these drugs.
So, what does that actually mean in scientific terms? The mechanism of action, or MOA for short, is the specific biochemical process that the drug messes with. It's the nitty-gritty of how one tiny molecule can stop a microbe dead in its tracks.
And here is the real magic of antibiotics. It's something called selective toxicity. They are brilliantly designed to go after targets that are unique to bacteria, like their cell wall, which our human cells don't even have. This lets them kill the invaders while leaving our own cellular machinery pretty much untouched.
So when you look at the big picture, most antibiotics work in one of these four main ways. Some stop bacteria from building that protective outer wall, the peptidoglycan layer. Others jam up their little protein factories, the ribosomes. And others prevent the bacteria from copying their DNA or making essential nutrients like folic acid.
Let's just zoom in on one of these to see how it works. Protein synthesis inhibitors are a massive, super important group of drugs. What they do is essentially starve bacteria by blocking it from making the proteins it needs to, live and function.
It's kind of like throwing a wrench into a factory assembly line. A drug like tetracycline will literally sit down in the spot where the next building block is supposed to go. And if you can't add new parts, you can't build anything. The whole factory just grinds to a halt.
Okay, so we've got our foundation, mechanism of action. Now let's add the second layer to our framework. This one's all about the drug's chemical structure. This is where all those family names you've heard before come into play. See, all the drugs that share a common chemical backbone get grouped together. And this is so useful because drugs in the same family often have similar properties, similar side effects, and even similar ways that bacteria learn to resist them.
Here's a perfect example of how our system nests together. Under that big category of cell wall synthesis inhibition, you've got these distinct chemical families. You have the beta-lactams, you have the glycopeptides, and others. They all attack the cell wall, but they each do it with a different-looking molecular tool.
Let's talk about maybe the most famous family of all, the beta-lactams. What actually makes a penicillin? It's this one core structure, the beta-lactam ring. This little four-sided ring is basically the chemical warhead of the molecule. It's the part that does the dirty work, binding to and shutting down the enzymes bacteria need to build their walls.
All right, so we've covered how it works and what it's made of. Now for the layer that really matters at the bedside. What bacteria does it actually kill? This is its spectrum of activity.
And look how beautifully our framework drills down even further. Inside the penicillin family, not all drugs are created equal. You start with the narrow-spectrum guys, like penicillin G, which are really targeted. Then chemists developed broad-spectrum versions, like amoxicillin, to cover more ground. And now we have the heavy hitters, the extended-spectrum drugs like piperacillin, which are designed to fight really tough bugs like Pseudomonas.
So why not just use a sledgehammer for every problem? Well, that power comes with a price. A narrow-spectrum drug is like a scalpel. It's precise and it causes way less collateral damage to the good bacteria living in our gut. A broad-spectrum drug is that sledgehammer. It gets the job done, but it can be messy. So the goal is always to pick the most targeted weapon you can.
And this isn't just some academic thought exercise. Choosing the right spectrum has huge real-world consequences. As Dr. Jeffrey Gerber points out, using a broader antibiotic than you need to doesn't just drive resistance, it means more side effects for the patient, more missed days of school and work, and real financial burdens on families.
This is a critical part of what we call antibiotic stewardship.
To round out our framework, there's one more useful lens we can look through, and this one gives us a little historical context. It's classifying the drugs by their source. Basically, where do they come from?
Really, the history of antibiotics is this amazing evolution from discovery to pure design. It all started with nature, right? Fleming and his moldy petri dish. Then we got clever and entered the semi-synthetic era, where scientists would take that natural molecule and tweak it in the lab to make it even better. And today we have purely synthetic drugs, created entirely from scratch in a lab.
So, we've walked through all the layers: mechanism, structure, spectrum, and source. Now, let's put it all together into one simple, powerful framework that you can actually use.
This is it. The whole system boiled down into 4 simple questions. The next time you encounter a new antibiotic, don't just try to memorize the name. Ask yourself, how does it work? What family is it in? What does it target? And where did it come from? If you can answer these 4 questions, you don't just know the drug, you understand it.
And this framework isn't just for learning about the past, it's for looking to the future. As we face this terrifying crisis of antibiotic resistance, which one of these layers do you think will be the most critical for the next breakthrough? Will it be about finding brand new mechanisms of action? Or maybe designing completely novel chemical structures to outsmart these evolving bacteria? The answer to that is going to shape the future of medicine. Thanks for joining us.